defoliants in Southeast Asia. Indeed, it is fair to say that it was the persistent attack led by Messelson that finally closed the doors of Fort Detrick and got the United States out of the biological warfare business. This culminated in one of the rarest of all honors. One morning, Messelson found his name listed by Nixon among the Big Ten of White House enemies. In retrospect, how could one imagine a more supreme and historic honor? Naturally, all these attainments have won for Messelson all kinds of honorary degrees, decorations, and awards. And of course, he's a member of the National Academy of Sciences, the National Institute of Medicine. He's a fellow of the American Academy of Arts and Sciences, and all other kind of honors have come his way. It's indeed a pleasure and privilege to present Matthew Messelson, who will speak to the subject, Chemicals and Cancer. Matthew Messelson.
Every time Carol introduces me, I have the same thought, which is I should quit while I'm still ahead. As you know, cancer is a very common disease. 17% of deaths in the United States are due to cancer, and the expectation Americans have at birth, at the present time, of contracting cancer is 25%, the difference being those who, contracting cancer, nevertheless die of something else. The elimination of cancer would add about 16 years to the longevity of those who die of it. There are two general and not totally separate approaches one can take to the practical problem of cancer. One is to cure it, and the other is to prevent it. Nothing that I'll say tonight, so far as I know, has anything at all to do with curing cancer. And if it has anything to do with cancer at all, it has to do only with preventing it. There is a great deal of evidence that the variation in cancer rates from country to country, from time to time, from group to group, that this variation is determined by environmental variables, the alternative being genetic variables. Let me show you some of this kind of epidemiological evidence that environmental variables account for most of the variance in the frequency of cancer. First of all, considering geographic variation, cancers are generally named by the site of the primary tumor. It is true that a given organ is subject to a variety of different kinds of cancers that can be identified by microscopic examination and other techniques. But as a broad generalization, it's true that each organ is mainly afflicted with only one kind. And so as a rough terminology, it's possible to describe different cancers in terms of the organ in which the cancer first arises. So we talk of lung cancer, even though there are several kinds, of colon cancer, even though there are several kinds, and so on. If we look at the incidence of different cancers named by the organs which they begin in, from country to country, looking only at countries where the data are reliable, and making adjustment for the fact that the incidence of cancer is extremely dependent on age. For the majority of cancers, not all, it increases with age, and it increases very steeply as something like the fourth or fifth or sixth power of age. One can correct for this by determining the cancer incidence in each age group, and then calculating what the overall incidence would be in some model population, and usually the model population in the data I will discuss is the United States in 1960. So the first slide that I have, do I press the forward button? I'll try it. Yeah. Shows a list of several kinds of site-specific cancer. I guess you can see that. It doesn't look very bright to me.
It's okay.
Yeah, okay, fine. But I don't know what's on it until I can see it. All right. Well, I can't read it, but you can probably read it. And you can see here, this is perhaps esophageal cancer. I think that's the one that's up there. Yes, that's esophageal cancer. And in northeastern Iran, the risk by age, I think this is calculated to be the single risk incidence by age 70. That is, if no other causes of death existed, and that's easy to subtract them out if you know the specific rate at each age, the incidence of esophageal cancer in this part of Iran would be 20%. But in Nigeria, where it occurs at the lowest known rate, it's less than 0.1%, and so the ratio is a factor of at least 300. And for each of these site-specific tumors listed here, you see the country in which the incidence is highest and lowest, and the ratio of the two. And so there are very big variations in many of the leading sites of cancer. This is an example of the geographic variation. Now, one could imagine that this geographic variation has a genetic basis, although I think that idea would strike any human geneticist as most unlikely with such big numbers and such big differences between big numbers. Nevertheless, one can get at this question in a number of ways. One of them is to look at the incidence of cancer in people who have migrated from one country, a country of high frequency of a particular cancer, to one of low, or for other cancers, one of low to one of high, and see what happens in the first generation, or ideally in the second and third. Here are some numbers on the second slide. In which I have expressed the age-standardized mortality for stomach cancer, lung and bronchus cancer for men, intestinal cancer and breast cancer. I've expressed those age-standardized rates in Japan as 100%. And in the United States, they are different. Stomach cancer is much rarer in the United States, only 8% of the Japanese incidence. Lung and cancer of the bronchus is higher, 3 times, more than 3 times higher. In cancer of the colon and rectum is almost 5 times higher, and breast cancer is higher. These numbers are based on people who have been born in the United States of parents who have emigrated to the United States from Japan. The parenthesis means that the number of patients on whom this is based is rather small and not too reliable, therefore. But you see that in all cases, the incidence for people who are the offspring of Japanese people, but they are living in the United States and born in the United States, is intermediate, and in general has gone a long way towards the prevailing rate in the United States. So that is strong suggestion that there are important factors in the environment, not in the genes, that determine the difference between Japanese and United States tumor rates for these cancers. Another way of getting at this question of the importance of environmental factors is to look at different occupational or different social groups. There are some striking cases. For example, half of all the men who have worked in benzidine factories, approximately half, contract bladder cancer. That can't be genetic. We know that roughly 5% is the likelihood of dying of lung cancer if you smoke on the average a pack a day of cigarettes during your life, throughout your lifetime. 20% of asbestos workers will contract a form of lung cancer due to their exposure to asbestos, or something, almost certainly asbestos, but one could perhaps say something in their industrial exposure. These special groups, and one could go on with other special groups, are clear evidence that there are important environmental factors at work. A much broader kind of look at this question comes from looking at site-specific tumor rates as a function of time. And this is an interesting subject because we hear sometimes that cancer rates are going up, or cancer rates are going down, or they're staying the same. And all those statements are true. It depends on the kind of cancer. The overall rate is climbing, but very, very slightly. If you look at the incidence, in this case for United States males, of different types of cancer, beginning, and these incidence numbers are age standardized, of course, to correct for the changing age distribution of the United States population. This is the year 1930, and this is the year 1970, and there have been some slight reclassifications, redefinitions of these different types of cancer, and these curves take that into account. One sees, of course, this enormous increase in the frequency of lung cancer, almost all of which is associated, statistically, with the increase in cigarette smoking amongst men, with the time lag. There is, on the other hand, a very steep decline in stomach cancer. See how it goes down, an enormous decline. If we look at the situation for, and then there are other kinds. Oh, by the way, I might use this slide to ask a question of interest. The use of synthetic organic chemicals in our society cannot be dated at any particular date, but the enormous use could be dated at the end of World War II. For example, before World War II, although some organic pesticides were used, very little were used. But after World War II, first with DDT and 2,4-D, and then with many other compounds used very widely, there grew up an industry which is now enormous in size and which exposes us to a very large number of different substances, in this case pesticides, and in various concentrations, but ranging up to concentrations of the order of milligrams per kilogram of us. Nevertheless, one doesn't see any inflection points that correspond with the end of the Second World War. There are some things which are rising, take lung cancer, but that rise started before, long before the war. Pancreas cancer is rising, but the rise started, you can't see it quite so well on this plot, but the rise started well before World War II. There are no upward inflections which begin after World War II, and some comfort could be drawn from that, but on the other hand, as you'll see later, when I discuss the age dependence, which I've already hinted at by saying that it goes as a high power of age, and as you'll see a little later, that it is not age that matters, but rather the duration of exposure. We haven't had very many years of exposure since 1945, in the sense that for many of these things, the steady state concentration has not been built up until some years after that. These statistics only go to 1970. Statistics to 1975 are not yet available. I'll discuss that question of whether there are any major changes going on due to the growth in the chemical industry later. But this plot at least doesn't show any evidence for such changes post-World War II. If we look at the similar data for United States women, we see something very different in the case of lung cancer. It's very low. But here it's beginning to rise steeply, and in the last few years that rise has continued almost superposably with the rise in men several decades earlier. And again, almost exclusively seems to be associated with the smoking of cigarettes. Again, the stomach cancer rates coming down in women. Breast cancer about the same. The overall cancer rates, as I mentioned, are staying approximately the same, a very slight rise. Sorry. Okay. Nevertheless, to get back to the point of environmental factors, it's obvious, I think, that the genetic composition of the United States public has not changed anything like enough to give these enormous changes in the large incidences of the primary sites of cancer. So it's clear, I've probably labored the point too much, that a great deal of the variance in the cancer rates is determined by environmental factors. It is of interest to ask what would happen if we took the lowest site-specific incidence for every site of cancer from the country in the world where it's lowest, and then added all those up and asked what would be the total as compared with the total in the United States. And the answer, if you do that, is that the total would be about 1/8 of what it is in the United States, so that if we had the knowledge and also the socially effective means of manipulating the environment in whatever ways might be needed, that would suggest that we could aim for as much as an 8-fold reduction in prevailing cancer rates. Actual differences between countries, by the way, are also of some interest. Countries where statistics are reliable, the highest overall cancer mortality rate is in Austria. Don't know why. And the lowest in Portugal. And the ratio is about 1.6. All right, so there are strong environmental factors. What are they? They could be music. Just because the title of the talk is Chemicals and Cancer, I can't leap to the conclusion that all these environmental factors are chemicals. Probably they're not. But there are only a few things that have been thought about, music not being one of them, of course, but viruses are. A very great deal of work has gone into trying to find viral causes of human cancer. Now, this work has been successful not with humans, but with some other animals. There's very clearly a disease, feline leukemia, another bovine lymphosarcoma, caused by viruses transmitted horizontally, that is, from animal to animal, not through the parent-child, not through the germline, but from animal to animal. They can cause a high incidence of these two diseases, and yet there is no clear case in man. There may be some association between some of the herpes viruses and certain particular cancers in man, but as yet there is no conclusively demonstrated viral cause of human cancer. More work may reveal such viral causes. Considerable amount of work has been done. There is no clear-cut proof of a viral involvement yet, though it's hard to believe that animals will be totally different from people. Impossible to believe. I would expect there will be some viral cancers found in people. But we do know a great deal about chemical carcinogenesis in animals and in people. First of all, in animals, it is possible to induce all known kinds of cancer chemically. Leukemias, lymphomas, sarcomas, carcinomas, cancers of all the different major sites of cancer, all can be induced by particular chemicals. That's interesting because the viruses so far, even in animals, don't induce all kinds of cancers, just a limited number of kinds, and principally cancers which are not of the epithelial tissues, which are the main cause of human mortality, cancers of the epithelial tissues. Chemicals in animals can cause not only all kinds of cancer, but they can cause cancer whether administered early or late in life. And in man, there are many chemicals known which cause, well, I shouldn't say many because the list is only about a dozen, but there are about a dozen chemicals known to cause cancer in man. The list for animals is much longer, but we ought to expect that difference because we get this information in man only incidentally from studying accidents or accidental exposures or unintended exposures and by difficult sifting out of epidemiological information. But the next list shows you the compounds or materials which are known to be carcinogenic in man. I've left out things of highly mixed nature like coal tar, which is certainly carcinogenic, but it's a complex mixture. You could accuse me of not being consistent by including asbestos, though it's a little pure due to its crystal structure. Here's aflatoxin B1, 4-aminobiphenyl, arsenic, asbestos, auramine, an intermediate in the dye industry, benzidine, bis-chloromethyl ether, chlornafazine. I should say what these are used for. Aflatoxin B1 is a fungal product formed by Aspergillus flavus, hence its name, in spoiling grains, husks of peanuts, some other places. The evidence for its being carcinogenic to man comes from populations of humans who have a high incidence of a special kind of liver cancer and an unusually high exposure to aflatoxin B1. In other words, it's an association, not an experiment. And the association, nevertheless, is strongly suggestive that the aflatoxin B1 causes liver cancer in man. because in different communities, the actual amount of aflatoxin B1 exposure correlates with the incidence of the adenocarcinomas of the liver in Thailand, Kenya, Madagascar. 4-aminobiphenyl, industrial exposure, men get bladder cancer from it. Arsenic, human carcinogen, lung and skin cancer. Asbestos, I've mentioned before. Auramine, used to be used in the dye industry. Benzidine, I've mentioned before. Another chemical intermediate, industrial chemical intermediate. Chlornafazine, a drug administered to patients for polycythemia vera in Scandinavia, no longer used, causes bladder cancer, 20% of the people who got it. Cigarette smoking, we all know about. Diethylstilbestrol, which clearly causes vaginal and cervical clear cell carcinomas in the daughters of women who received it during pregnancy. 2-naphthylamine, industrial compound, causes bladder cancer. Mustard gas, Japanese workers making mustard gas in World War II, high incidences of lung cancer. Vinyl chloride workers get a rare liver adenocarcinoma. You notice that for all of those but one, that is asbestos, the chemicals are also carcinogenic in animals. Of course, the whole practice of testing things on animals to see if they're safe or not would rest on the reverse question, namely, are things which are carcinogenic in animals carcinogenic in people? Asking the question in that direction we cannot do directly because you cannot do experiments on people. But the reverse question, the answer is quite clear. Nearly everything known to be carcinogenic in man is carcinogenic in rodents. The exception possibly being arsenic, but the evidence isn't fully in yet on arsenic. Okay. So chemicals certainly can cause cancer. How might they cause cancer? An old suggestion is that cancer arises because of mutations, genetic changes. Bovary in 1914 suggested that, not due to chemicals. In fact, chemical mutagenesis wasn't known then. And it has been a prevailing hypothesis simply because the cancer cell gives rise to more cancer cells and therefore behaves like it's a change in the inheritance of the cells. So it's natural to suggest that cancer is due to a mutation. Whether it really is due to mutations, changes in the structure of DNA, we still don't know, but that is one of the most promising hypotheses, as I will discuss. So the thought that cancer is caused by some kind of mutation is an old thought. The thought that mutations can be caused by chemicals is a newer thought. During World War II, a British physiologist, Robeson, studying mustard gas under military secrecy, observed that mustard gas causes changes in the appearance of the nuclei of cells that look like the changes caused by X-rays. And Muller, before World War II, had showed that X-rays cause mutations, and Robeson then collaborated with a young Drosophila worker, Charlotte Auerbach, and during the war demonstrated that mustard gas and a variety of other chemicals cause mutations. And subsequently, that work has expanded very greatly. We know that a large number of chemicals can cause changes in the DNA which are heritable through the germline. They are mutations. So this piece of knowledge that chemicals can cause cancer, and that's been known for a long time, it's hard to date it. Percival Potts, more than 200 years ago, noted in his book, that exposure to the tars can cause scrotal cancer. Experimental animal carcinogenesis was achieved in the 30s. So that fact, chemicals cause cancer, chemicals cause mutation, has led a number of people to try and put the two together to show that those chemicals which are mutagenic are also those chemicals which are carcinogenic and vice versa. And a lot of people have tried to show that. Tatum and Demeretz and many of the founders of modern microbial genetics took a try at this, and the results generally were suspiciously encouraging but never very good. The reason they were never very good was suggested by Boyland in 1947 in Britain, and that is that a lot of compounds which cause cancer, when administered to animals, are metabolized by the body to become the real carcinogens, and by themselves are not carcinogenic, and therefore the suggestion would be maybe they're not mutagenic either. In other words, maybe in order to test something on a bacterium, which doesn't have the same kind of intermediary metabolism and catabolism that animals have, maybe you have to do something to the carcinogen that normally happens in the animal body to make it into a mutagen before you test it in a microbial system to see if it's mutagenic. I should have asserted with a little bit more emphasis that the standard ways of testing for mutagenesis are in microorganisms, being easiest. When this was realized, it set the stage for a new attempt to correlate carcinogenesis and mutagenesis. And this has been done most vigorously by Bruce Ames in his Bruce had been interested in mutagenesis initially, and then because of an interest in environmental toxicology, a sideline for him. He's a molecular biologist, and it's been said, and I would agree with the statement, that this is probably the most practically useful contribution to come out of molecular biology yet, the so-called Ames test. Now let me describe this test. It's a very simple kind of experiment. One starts with the bacterium Salmonella, a bacteria very closely related to Escherichia coli. One starts with it because Bruce's work before that was with it, with a mutant of it which cannot make the amino acid histidine, and so it's called His minus. And you put about 100 million of these cells, of bacterial cells, on a petri dish. Petri dish is about that big. And on the petri dish you don't put any histidine, so those bacteria cannot grow. And if you put the petri dish in a warm room overnight and look at it the next morning, there won't be any colonies, except for the very rare bacterial cell which has mutated spontaneously to become His plus. Now on this plate, this petri dish, which has no histidine, the His plus bacterium can grow, and every half hour it divides and its descendants divide at the same rate, so that by morning one sees a colony. But the background rate, the rate at which that happens if you don't stimulate the mutation process, is low. There will be only 5 or 6 such colonies growing on that plate. Now there are certain chemicals which you could add to the plate, and they will greatly increase the frequency of revertance without adding anything else, just the chemicals. But most of the chemicals which are carcinogenic, which had been tested in systems like this, didn't make many more colonies, and therefore weren't seeming to be mutagenic. And the addition that Ames made was to add liver extract. The liver is rich in enzymes which activate many compounds to strongly electrophilic derivatives. So on this plate we add the 10 to the 8th bacterial cells plus liver extract, usually from rats, because it's easy to get. And now we can test chemicals. It turns out that a very large additional number of chemicals that didn't appear to be mutagenic when you leave out the liver extract now are very strongly mutagenic. For example, aflatoxin B1 is one of those. By itself, it's not mutagenic. Liver extract by itself is not mutagenic. Aflatoxin B1 and liver extract, very mutagenic. In fact, the most mutagenic substance known. Okay. So what Ames has done and others have done is to test lots and lots of chemicals, chemicals which also have been tested for carcinogenicity, and to look at the association. Now the selection of the chemicals is a problem. You could say, well, how do you know it's an unbiased collection? Well, the only real answer to that is time will tell. This slide divides some of the compounds he has tested, or actually all the compounds he had tested as of 2 years ago, into various chemical groups, which you can read better than I, alkyl halides. So what's important is the totals out of 100 and, well, I'll read you some newer numbers because I can't read the slide from this angle. But for example, his current numbers are that out of 175 substances known to be carcinogenic in tests on laboratory animals, 157 score positive in his test. That's 90%. And of 108 chemicals which, when tested in animals, have not convincingly given cancer, 95 of that 108 also do not give mutations in this test, and that's an 88% association. And now one can make excuses. Always when the agreement isn't perfect, one can make some excuses. There are some valid excuses, and then there's some cases where there are no valid excuses known. For example, there is a substance called cycazin, which is known to be quite carcinogenic in rodents, but it's not mutagenic in this test. But the answer turns out to be that there are bacteria in the gut which convert cycazin to another compound, and that compound is highly mutagenic. And if you put the right gut bacteria together with the cycazin before you put it on the plate, then it's very mutagenic. So that's not really an exception, though in these raw numbers I gave you it would be. And there are a number of cases like that, plus the fact that the mutagenic test is more sensitive than the carcinogenic test. And so most of those cases where something shows mutagenic but isn't carcinogenic so far as has been determined in mice and rats, it's probably because the stuff is too weak to give a response in mice and rats at the doses tested. Nevertheless, there are some cases where there are probably exceptions. Sodium azide is hard to see. It's been given in huge doses. It is not carcinogenic to mice and rats, and it is mutagenic. Nevertheless, the association is very close. Does this mean that mutations are the cause of cancer? No. What it means is that chemicals which have the ability to mutate salmonella have the ability to cause cancer, and vice versa for the negative statements. But it could be that the actual chemical reaction which is causing the cancer is not with DNA. It could be with some very important protein. However, the working hypothesis of most people in the field at the present is it is in fact a mutation because of this transmissible behavior from cell to cell of cancer, even though one has to keep his mind open to the possibility that there could be semi-steady states in cells that really aren't based on changes in the DNA that might propagate like heritable changes. And that's a long and, at this point, not essential discussion for us. One might ask not only about the qualitative results from the Ames test. I might mention one other qualitative result, and that's an interesting one from the Imperial Chemical Industries in England. They have taken 120 compounds, 60 carcinogens, and then 60 chemical derivatives of them, compounds that are very closely related but derivatized and known not to be carcinogenic, and tested this group of 120 compounds by the Ames test. And there the success rate turns out to be 95%. That is, and it's about equally wrong when it's wrong in either predicting carcinogenesis when none is found or vice versa. So again, a very high correlation. What about the quantitative relationship between the strength of a mutagen for causing the change from histidine minus to histidine plus and the strength as a carcinogen? Now, I've been looking at that a little bit for the following reason. As I mentioned before, we're mainly at present at the mercy of animal tests in order to determine whether or not substances in our environment are carcinogenic. The FDA's decision on saccharin is based on tests in rats and earlier tests in mice too. We have this evidence that things that cause cancer to people, almost all of them cause cancer in animals, but it's the reverse statement we're more interested in. We can't get that information. Is there anything we can do? Yes, we could at least test the hypothesis that only the strongest carcinogens affect people and a whole bunch of other ones affect mice and rats. So we could ask about the strengths. And if we can rule that hypothesis out, it could still be that rats and mice are sensitive to lots of things that people aren't, but at least that simple way in which rats and mice might be sensitive to things that people are not, just a quantitative difference, could be tested. So it's of interest to try to define a carcinogenic potency. Let me define the carcinogenic potency K, for carcinogenic potency, as equal to, now, having defined it for a reason of convenience in a certain way, I'd better not change it. And it's totally unimportant, but I have to write it down to keep consistent with other things I've done. So I have to write log 2 here. which is of no importance, it's only equal to 70%, forget it. It might as well not be there. The definition of the carcinogenic potency is the reciprocal of the number of milligrams of the chemical in question that must be administered per kilogram of animal every day for a lifetime, that is, for the normal lifetime, and in rodents that's 2 years. So let's say this is for rodents, for 2 years. That gives 50% tumor incidence by the end of those 2 years. So you can see that if you have to give a lot of a chemical to get that much cancer, then it's not very strong. And that's why I've defined this as the reciprocal. So that's just an operational definition of carcinogenic strength. Easy to measure in the laboratory on mice and rats. I define a mutagenic potency, M, analogously, in a way purely of convenience, and that is the reciprocal, and here there's no log 2 to worry about, which you shouldn't worry about anyway, and that is the number of micrograms per petri dish, which in the Ames test gives 100 revertant colonies. A purely operational test. And you can see that if you had to put a lot of micrograms on, wouldn't be a very strong mutagen. The reciprocal of it, therefore, measures the strength of the mutagen. And you might wonder, why did I use micrograms here per petri dish and milligrams per kg? The reason I did was because there is a relationship, and the relationship comes out to be K equals M if I use micrograms here instead of grams or kilograms, or very closely equals M. At least for the few compounds for which I have made any comparisons. Now, Ames and his colleagues have determined the mutagenicity, as I mentioned, of 170, well, I don't have the exact number here, but it's over 100, almost 200 mutagens. Now, some of the things they've tested aren't mutagens, so we can't ask the relationship when the compound isn't a mutagen. I'm going to confine this comparison to compounds that are both mutagenic and carcinogenic, which is most of them. If a compound is one, it is the other, as I've mentioned. Now, the world literature on carcinogenesis testing is gigantic, and most of it is inappropriate for determining this value of K. The data are not presented in most publications so that one can do this. What I've done is to take only those compounds for which Ames has reported the mutagenicity in a particular publication of his. I chose that so that I wouldn't have any further choice over what compounds, because I could otherwise be biased. Bias could creep in. I took for the carcinogenic data only those data reviewed by the World Health Organization in a monograph series they published called the IARC, International Agency for Research on Cancer, Reviews of Carcinogenesis. And I took only those compounds that had been, on the one hand, published by Ames in his paper at the end of 1975, and on the other, reviewed in the first, well, in the then available volumes, 1 through 7, of the IARC monographs. And surprisingly, that knocked the total of 200 down to 27, which was good. Furthermore, it did the job of looking through the world literature, because the reviews of the IARC do that. But of these 27, some had been given only once, a single injection, and my definition of carcinogenicity can't work with single injections. You have to give it all the time, steadily. And I haven't even looked at what you can do with single injections. And if you consider only the tests where the exposure is chronic, and I also excluded skin application, because I don't know how you determine the dose with skin application. Then it boils down to 14 compounds. And for 10 of these, I have plotted the calculated value of K from the carcinogenicity versus the Ames value of the mutagenicity. And the plot is a log-log plot because the numbers span a large range. Actually, what I've plotted here is not K and M, but their reciprocals, the things you actually measure. The number of micrograms that you have to put on a plate to get 100 revertants, or the number of milligrams per kilogram per day that you have to give to get 50% tumors at the end of the life. Here's aflatoxin B1. There are 3 points because there are 3 experiments, all with rats, and they're connected by a line. Steric-matocystin is another fungal product. Benzoapyrene, as you know, a cyclic aromatic compound. Where there are lines between points, it means that they were different experiments, sometimes on different species. And these include all the experiments in the world literature for those compounds which meet the criteria of running long enough. And one sees that over this very large range, it's 5 log cycles, so it's a range of 100,000, that there is a fairly good correspondence between the mutagenic strength and the carcinogenic strength. Plotted this way, the most intense carcinogen is near the bottom, aflatoxin B1. It's also the most intense mutagen. The greatest deviation of any point is this one. It's a factor of 20 off the curve, and the average deviation is a factor of about 3. Recently, Ames discovered that there is a substance used to retard the combustion of children's sleepwear, which is positive in his test. And this substance, the common name of which is tris, it's tris-2,3-dibromopropyl phosphate, a simple compound, is positive in his test. And because of that, the tests that were going to be done on rats and mice by the government were accelerated to get the results out sooner. And sure enough, it turns out to be carcinogenic in mice and rats. And it was, we used this curve to predict how carcinogenic it was because we first knew that its mutagenicity was about here. And it turns out that that point falls right there on this curve. So it's made one prediction. It's a long way to go. Oh, actually, there's one more recently that's way up here. So I won't go into it. But I haven't seen the papers on that, but Ames told me about that one. So there seems to be, at least for these compounds, a good quantitative correspondence. And considering all the many variables that I will not have time to mention, this is a surprisingly good quantitative correspondence. But I told you there were 14 compounds, and there are only 10 there. There are 4 that I left off. I left them off because they didn't fit. But they're all compounds with nitrogen-nitrogen bonds. They're all nitroso compounds. And these compounds universally score low in the Ames test. They don't seem to cause the frequency of mutations that they ought to if this relationship were true. Maybe they just won't obey a relationship like this. But Ames has developed his test already by a lot of jiggering. For example, he's weakened the bacterial membrane so that things can get into it more easily. And he's done some other things. And he's now, and that's, after all, fair and square. If something didn't work in his test and it was merely because the molecule couldn't get into the bacterium, that's not of any theoretical interest at all. He's working on the possibility that the bacteria may have enzymes which very rapidly degrade nitroso compounds, and that those enzymes might be the reason that nitroso compounds don't score. In any event, they do not score very strongly. Nevertheless, turning to a practical application of this test, or let me make a thought application first and then tell you about a real one. If someone had suggested that an interesting leaf could be enjoyed when smoked, and the government were using the Ames test to find out if it ought to be encouraged, tobacco tar would be tested. And it turns out that tobacco tar is immensely mutagenic in the Ames test. Tobacco smoke condensate is immensely mutagenic. One would have predicted, whether correctly or incorrectly, one wouldn't have known at the time, but one would have predicted that cigarette smoking might be harmful to your health. A real prediction, which hasn't yet been tested, comes from what you might call the naive biochemist's approach to using the Ames test for dealing with a cancer problem. Now, one could ask, what if we had data for human carcinogenicity? Now, this is all rats and mice, but what if we had data for people? Would a similar curve result? Well, my guess would be yes, but it might be displaced. My guess would be yes just because there are no fundamental biological differences we know of between people and rodents with regard to those things that have to do with the cellular biology of cancer. But the curve might be displaced. What units do we use to express dose? to convert dose in humans to dose in rats and mice, or to express time. Well, let us make the supposition that time ought to be measured not in years but in lifetime, simply because nature has rigged things so that both mice, rats, and we all get 20, 30% cancer a little while after the reproductive period is over. It's as though the clock that nature is using, in a very crude sense, is not years, but lifetime, which for us is 70 years, more or less, and for rats only 2. So if we just change this definition to 70, let that be the definition for K for man, instead of for rodents. So now we have K for rodents defined by the expression I had there before, M, the mutagenic potency, and K for man. I've asserted that for at least some compounds there's encouraging suggestion that K equals M with rodents. What about K for man and M and K for rodents? Well, there are very few points, but one of them is cigarette tar. Guinea pigs who have been given cigarette tar by inhalation do develop cancers very similar to the kinds of cancers people get, and the incidence can be measured, and in that case it turns out that the K value for the rodent, in this case guinea pig, and for man are very close, within a factor of 3 or 4. Another case is aflatoxin B1. For aflatoxin B1, one knows roughly the human intake in the countries I mentioned, Kenya, Thailand, Madagascar. The average intake that gives, well, I won't go into this in great detail, but let's write down some numbers here. The epidemiology of adenocarcinoma of the liver in these countries suggests that you get somewhere between, these are 90% confidence limits, I believe, between 0.02% and 0.1% liver cancer for lifetime exposure to aflatoxin B1 when you get about 1/10 of a microgram. That's 10 to the minus 4 milligrams per, I guess, about a 50 kilogram person averaged over life in both sexes in people living in these countries, who knows, not too important, per day. Now if K equals M, the mutagenicity of aflatoxin B1 is 200 on this definition, and so is its K value for rodents, because it just about fits that curve. Now if we put 200 in this expression, we predict that the incidence would be 0.04%. That may be an accident, but again it comes within the range of estimates from the epidemiology of adenocarcinoma of the liver in humans. So let us say yes, at least tentatively, that the human curve is the same curve so long as we measure time in units of lifetime instead of units of years. If that's the case, then we could say, what would be the number of revertants on the salmonella plate if we took a quantity of a carcinogen which, given every day to a person, caused a 1% lifetime cancer incidence? And you can grind through these little equations and find out that if you could get all of the chemical that hypothetically causes 1% incidence of cancer in me, put it on that dish, and it would give me 17,000 colonies. Well, that's a lot. What that means is that the test is sensitive, which you already knew, because I told you that it could pick up cigarette smoke, and it tells you something. What that means then is that there's hope for essentially fractionating the things in people, like biochemists do to find enzymes. The assay being the Ames test, to see if you could purify, isolate, and identify the prevalent chemical carcinogens in us. You grind up people, run the extracts on columns, get 1,000 fractions, test them all. One is very active, fractionate it into 1,000 fractions. One is very hot, hotter than anything before. Test it until finally you get a pure compound, determine its chemical structure, and then wonder about whether or not you can reduce the exposure. This approach is being taken in a number of laboratories, and there's a very interesting result from the laboratory of Dr. Robert Bruce in Toronto, Canada. Now, he has not been grinding up people. He has been looking in feces for chemical carcinogens. He has been looking at a number of individuals and finds that in some individuals in the feces there is an enormously powerful mutagen, equivalent in the amount excreted per day of this mutagen to the mutagenicity of about 3 or 4 packs of cigarettes. So that's very strong. Now, this compound is particularly interesting. It turns out to be a nitrosamide, although just what nitrosamide, I don't think he knows yet. In any way, I don't know yet. So that means the Ames test is probably underscoring it, if anything. It shows a very interesting difference between individuals. Some people produce a lot of it. Everybody produces some of it. Some people produce 5 times more than other people. He has analyzed the dietary intake, and none of this can be detected in anything you eat. So this is an example, apparently, of a chemical, but it is not a chemical which itself is taken into the body. It looks more as though it's a product of metabolism, or perhaps a product of the flora of the intestinal tract. The compound doesn't seem to depend on diet. He's taken individuals who have high levels and low levels and tried very hard to trade diets. Now, I know that that can be tricky because they're cheaters, but he finds no effect of trading diets. People who are vegetarians, meat eaters. The gentleman in question who has the highest incidence of this material is a meat eater. One of the low people is a vegetarian, but by trading diets, there's been no effect in the output. So whether or not this is responsible for the enormous frequency of colon cancer in the United States, which, as you may have noticed, is not only very high but rising, remains to be found out. That will take a lot of very detailed epidemiologic comparisons. But it is the beginning of an altogether new approach, a brute force approach. Now, there's a big word of caution that must be noted, and a lot of things that I'm going to have to leave out. The word of caution is that we know that cancer is not a one-step proposition. We know this partly from the age incidence curves. If you plot the incidence of cancer, well, let me show you the next slide. You'll see that. Here are some log-log plots of the incidence of cancer plotted on the vertical axis for particular kinds of cancer, pancreas in males, pancreas in females, and so on, stomach, males and females, against the logarithm of the age. The fact that these lines are very straight means that the incidence of cancer, the number of people per unit time, divided by the size of the population, so that's the incidence per unit time, has the form of, it's negative because people disappear when they get it, has the form of minus a constant times time to a power, and the power is of about 5. The log-log curve being a straight line means that it fits this sort of relationship. And that suggests, it doesn't prove, but that suggests steps. That suggests that there are a number of steps involved in getting cancer. 6 steps if this were 5 and on some of the simplest models. But there's more direct evidence that there are different steps. The chemicals that score plus in the Ames test and are carcinogenic in animals, still, at least some of them, by themselves are not very carcinogenic. For example, methylcholanthrene is carcinogenic in the Ames test, and if you paint the back of a mouse with it, you'll get skin cancers. But if you paint the back of a mouse with methylcholanthrene and then afterwards paint the back of the mouse with something that's not at all positive in the Ames test, namely an ester of Forbol, a complicated heterocyclic compound found in croton oil, you get an enormously larger crop of tumors. And you don't have to follow the methylcholanthrene treatment immediately with the Forbol ester to achieve this promotional effect. You can follow it a year later. So there's and if you do it in the reverse order, there is no synergistic effect. It has to be methylcalanthine, a mutagen, first, 4-ball ester, not a mutagen, second. The effect is called promotion. The 4-ball ester is called a promoter. It hasn't been much studied in any other systems, but there are other things like that. You can give radiation, which is mutagenic and carcinogenic, to animals, and you'll get some thyroid cancers. But if you give thyroid-stimulating hormone later, and not immediately later, but even much later, you get an enormous increase. So this suggests that there are steps. Step 1, take cells to some new state. Step 2, which only after a number of steps gives you the result of cancer. And that the mutagens are somewhere near the beginning, and we're beginning to learn quite a bit about those, and that there are other substances that may intervene later, that may act like hormones to stimulate division, to release cell division. It's not really known. Much less is known about promoters. Now, to what extent now, if this were always true, and one could always remove the things that cause step 1, one could still have a perfectly effective method of reducing cancer rates. But if it turns out that some of these substances are not manipulable, either for social reasons, cigarette smoking we've known about for a long time, and yet the incidence of smoking does not go down. So there's a social reason why, even though we know what it is, it hasn't had the slightest impact, or at least it hasn't had a major impact on the incidence of the disease or the prevalence of smoking. But if there are a number of steps, it may be that it will turn out for some cancers to learn how to identify these compounds, of which much less is known. And that's the big caveat I have to make in this talk, is that one mustn't get the notion that mutagens by themselves are the sole stimulate a single step which then gives cancer. But like a chain, the removal of one link can nevertheless break the chain, prevent the end result. So, to summarize, most of the variation in cancer incidence worldwide, in time, occupation to occupation, most of this variance is due to environmental factors. If these factors could all be controlled simultaneously, we could reduce cancer incidence by at least a factor of 8, maybe more. There's a strong suggestion that exposure to carcinogenic substances of both external and internal origin causes a large proportion of environmental variation in cancer rates. Now, that large proportion statement, I can get there from just cigarette smoking and Bob Bruce's result. Cigarette smoking already causes more than 30% of all the cancer in England. Not quite so much here, but still very nearly that amount. If Bruce is right and one adds colon cancer, then there are 2 cases where it's substances and not viruses and not music. So one would be able to say, with those 2 things alone, one ought to be, if only one could rid the environment of them, be able to reduce the major part of the cancer incidence. And presumably other discoveries lie ahead. These substances that do this are both carcinogen mutagens, things that can be detected in the Ames test, and also probably so-called promoters. And the relative importance of these 2 in causing the observed variance in the cancer rate, that's not known. The relative degree to which they will be socially manipulable, that's not known. But we can draw some comfort from the fact that if this picture is right, either one that you remove still blocks the final outcome of the development of the disease. And thank you very much.